Alright, hi everyone. Today we're going to cover the 2022 May paper 1R, as you can see here. This is the IGCSE Human Biology paper, and this paper has a total of 90 marks, and it will test you on the entire syllabus. So let's start off with the first question. Question 1, the diagram shows a human body cell. Name the parts X, Y, and Z. So X here is the cell membrane. So the cell membrane is responsible for controlling substances that come in and out of the cell. Y would be the nucleus. So that stores the genetic information, and Z is the cytoplasm. Okay, name one structure in the cell that is not shown in the diagram. So there are plenty of things that you can say about this. You can say mitochondria, which is responsible for producing energy through the process of respiration. You can also say ribosomes as well, and you can also say endoplasmic reticulum. So I'm just going to write mitochondria here. Mitochondria. Okay, now I'm going to show you a diagram for this part. So here's a diagram that I've included. So an organ, which is this part here, an organ consists of different tissues working together to form a specific function, and a tissue are basically similar cells that are working together to serve a particular function. So you need to write all those 2 points down. Let me just make this one smaller. Okay. So organs are made of different tissues working together and a tissue consists of similar cells. Working together. Okay, let's move on to part 3. Part B, the boxes list 3 processes that transport substances in and out of the cell. Complete the diagram by drawing one straight line from each process to its correct definition. So osmosis would be the movement of water from high water potential to low water potential. Diffusion would be when molecules move from a high concentration to a low concentration. And active transport would be against the concentration gradient, and this would require energy. All right, let's move on to part C. So for part C1, the diagram shows 2 types of epithelial cells, A and B. Give the name of each type of epithelial tissue. So tissues that line organs are referred to as epithelial cells or epithelial tissue, and there are 2 types that you need to know for your syllabus, that is squamous and ciliated. So squamous, you can kind of tell by the name, it just means that they're sort of flattened, and you can see this in the linings of your cheek. And the ciliated epithelial cells, which is this one here, so this is squamous on the left and epithelial on the right, and ciliated on the right. These ciliated epithelial cells, they have these things called cilia, and these allow for these cells to brush away, for example, mucus away from your lungs. So let's write this down. So A would be squamous and B would be ciliated. Let's move on to part B. An epithelial cell has an actual width of 60 micrometers. The cell is magnified 40 times. Calculate the image width of the cell in millimeters. Equation for magnification is magnification equals image size over actual size. So let's rearrange the equation so that the image size is the subject of this equation. So that'll be magnification times actual size. So the magnification here is 40 and the actual size is 60. So if we times those two numbers together, that would give us 2,400. However, they want you to give this answer in millimeters, and we can see here 1 millimeter equals 1,000 micrometers. So because of that, so this is in micrometers, we need to convert that into millimeters by dividing it by 1,000. So this divided by 1,000, that'll give you 2.4 millimeters. All right, 2.4. Question 1 done. So question 2, the diagram shows the human digestive system. So here we have a person. Let's label the different structures. Let's start off with X first. So X is the esophagus. So I'm going to use a British spelling for this, and that starts with the letter O. So esophagus. All right, then after the esophagus, the food will go down to the stomach. So I'm going to write stomach here. Next, it goes down to the small intestine, so that is Z. So that is the small intestine. And then W here, we have the large intestine. Okay, let's move on to the next part. Part 2. The box gives words used to describe how food is moved through structure X to word structure Y. Use words from the box to complete the passage. Food forms a bolus at the back of the mouth. That would be bolus. So bolus is what we refer to the food once it's in our mouth and mixed in with our saliva. The food is swallowed and passes into structure X. So structure X, as you can see here, that would be the esophagus. So the walls of the esophagus, or the walls of structure X, contains something to push down the food towards Y. So this is referring to peristalsis. And peristalsis is a process in which muscles kind of they contract to sort of push the food down, kind of like when we squeeze toothpaste out of its container. So structure X contain muscles which contract to push the food towards structure Y. This process is known as peristalsis. Okay, let's move on to part B. Okay, part B, the diagram shows some apparatus used to investigate the effect of an enzyme called amylase on starch. So just to do a little bit of recap, starch is what we refer to as a polysaccharide. So poly means many, and starch is basically many glucose molecules linked together to form a larger molecule, and we refer to that as starch. Right, and amylase is responsible for breaking down that starch into smaller sugars. So here we have, in the beginning, we have the amylase and the starch solution inside of this Visking tubing. And what would happen is that the amylase would break down the starch into smaller sugar molecules, and what would happen is that the sugar molecules are small enough to actually diffuse out. So they're able to diffuse out. However, the starch would remain inside of the Visking tubing because they're too large enough to pass through the pores in the Visking tubing. Okay, let's look at part 1. Name one area of the digestion system where amylase is produced. So there are two places that you can list. You can say number 1, the salivary gland, or number 2, the pancreas. So I'm just going to write salivary gland here. Salivary gland. Okay, name the structure in the digestive system that the Visking tubing represents. So amylase breaks down starch to simple sugars in the small intestine. So that's where it takes place. Small intestine. Now let's look at part 3. Explain the results of the investigation after 30 minutes. So I've already just mentioned it previously. So amylase breaks down starch into sugars. The sugars then diffuse out through the Visking tubing. Okay, name the chemical used to test for sugar in the water surrounding the Visking tubing after 30 minutes. So it's referring to this darker liquid here. So in order to test for the presence of sugar, we would use Benedict's solution, or you can say Benedict's reagent, that's fine too. All right, let's move on to part 5. After 30 minutes, iodine solution is added into the contents of the Visking tubing. So that is inside of the Visking tubing. Now iodine tests for the presence of starch. And you can see here, in this diagram that I've drawn, the starch will still remain inside of the Visking tubing. So the original color of iodine is orange brown, you could say that. And in the presence of starch, it'll turn blue black. So let's write that down. The iodine solution will turn blue black because starch is present. Okay. So those are the 2 marks. Another mark that you can also say is this is because starch is too large to pass through the Visking tubing. All right, let's look at question 3. So part 3, or question 3, in England, 3,319,266 people have diabetes. Give this value to 3 significant figures. So you would just write the 3 sig fig here, but we would round this 1 to a 2 because this next number here is greater than 4, so it's 5 and above, right? So we would write 332,0,0,0. So that would be the answer for that. Next part. Give in the rest of the United Kingdom, 600,239 people have diabetes. Give this value in standard form. So standard form would be something, something, something times 10 to the power of something, right? In this case, what we would do is imagine I have, let me just zoom in. Imagine if I had a decimal point here, right? So I would move this 1, 2, 3, 4, 5. Therefore, it'll be 6.00239 times 10 to the power of 5 because I've moved the decimal point 5 spaces. This is how I figure things out. Okay, let's write that down. So that'll be 6.00239 times 10 to the power of 5. Okay, let's move on to the next part. Diabetes can be caused by the inability of an organ to produce insulin. Name that organ that produces insulin. That would be the pancreas. Okay. All right, let's move on to the next question. The body mass index, BMI, is a measure that uses height and body mass to work out if the body mass is healthy. The table shows the BMI values and the categories into which a person can be placed. So it says here, calculate the BMI of a person with a body mass of, let me just highlight this, whoops, 120 kilograms and a height of 1.95. So let's just plug in the equation or plug in the numbers. So 120 divided by 1.95 to the power of 2. So let me just use the calculator here. To the power of 2. And here we get 31.56. So 31.56. Okay. Part 2, determine the BMI category that this person belongs to. So 31.56, this would be in this category here because it lies between 30 and 39.9. So it'll be the obese category. All right, let's move on to this next part. So part C, the graph shows the relative risk of developing a form of diabetes called type 2 for different BMI values. So here are the BMI on the x-axis and then the relative risk of diabetes on the y-axis. Describe the trend shown by the graph. So usually we would phrase the answer as this: as something 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 increases, the other thing increases or decreases, right? In this case, as the BMI increases, the relative risk of diabetes increases too. So as the BMI increases, the relative risk of diabetes increases. Another thing as well is that in the lower BMIs, it's not really increasing as much, but after BMI of 24, that's when it starts to increase pretty rapidly. So you can say at around a BMI of 24 to 24.9, the risk increases rapidly. Oops, rapidly. Okay, let's look at part 2. Give some dietary advice that could help an obese person lower their BMI. So there are a couple things that you can say. You can say this person should eat a balanced diet, or you can say eat less fats or eat less carbohydrates or less sugars. So you can say a balanced diet and to eat less fats. Sorry. Okay, let's move on to question 4. Okay, part A, lipids are digested in the small intestine. Part 1, describe a test for lipids in food. So I'm just going to show you a little picture here. So to test for the presence of lipids, number 1, we would actually add ethanol to the food sample and we would shake it first. So the ethanol would dissolve the lipids, and then we would add water. And we would add water because water and lipids don't mix very well together. It forms what we call this thing here. It starts to become more cloudy or more milky. Okay, So let's make this one smaller. So you would add ethanol to the food sample and mix. Then add water. If lipids are present, it will turn cloudy. Okay, part 2. Name the enzyme that digests lipids. That would be lipase. Part 3. Name the products of lipid digestion. So fats will get converted to glycerol and 3 fatty acids. So you do need to remember these products. Okay, let's move on to part B. Okay, part B. A student investigates how the pH of a lipid solution containing an enzyme that digests lipids changes over 10 minutes at 2 different temperatures. So the temperatures are 37 degrees here and 80 degrees here. Okay, so the reason why we test for the pH is because when we break down fats, it forms fatty acids, and fatty acids are acidic, and that would cause the pH to decrease. Let's go on to the next part. Part 1, estimate a pH value for the lipid solution at 80 degrees at 5 minutes. So it's talking about this blank space here. So we can see that it is slightly decreasing, so maybe we can write 8.7 there. So we can say 8.7. They do accept a range between 8.6 and 8.8, so any number between these two numbers is fine. Part 2, give the independent variable and the dependent variable for this investigation. So the independent variable is what we're changing, and here we're looking at 2 different temperatures, 37 and 80 degrees. So here the independent variable would be temperature. Now, the dependent variable is the thing that we measure. So here, according to the table, we are measuring the pH. So that'll be the dependent variable. Okay, so like I mentioned previously, because the digestion of lipids forms fatty acids and that decreases the pH, we can see here that when it's at a temperature of 37 degrees, the pH went from 8.3 all the way to 5.5, so that's a pretty big decrease there. Whereas here at a temperature of 80 degrees, it goes from 9.2 to 8.4, so there's not much of a decrease there. So we can say it looks like the 37 is closer to the optimum temperature, or is the optimum temperature, and that's why the lipase works faster. Whereas 80 degrees, that's when the enzyme is denatured. So let's write that down. At 37 degrees, the enzyme is working at its optimum temperature. So the reason why it's 35 is because that is our body temperature. Optimum temperature, which is why the enzyme activity is greater. However, at 80 degrees, the enzyme has denatured. So denatured means that the enzyme changes shape, and because the enzyme changes shape, then the substrate is no longer able to fit into the active site. All right, and that will be the 3 marks. Oh, it's asking for 4 marks. Okay. So we can say that, which is why the enzyme activity is greater. Therefore, the fat is digested more quickly. So the 4 marks are 37 degrees, optimum temperature, that's 1 mark. Second mark is enzyme activity is greater. Third mark is fat is digested more quickly. Fourth mark is at 80 degrees, the enzyme has denatured. So those are the 4 marks. Okay, let's move on to question 5. Great. Question 5. The table gives information about 2 methods of contraception. So the question is asking, evaluate these 2 methods of contraception using information from the table and your own knowledge. So let's go over these 2 methods. So male condom is what we refer to as a barrier method. So that is very good at preventing against STDs or sexually transmitted diseases. So that's a barrier method, where sterilization is when so this could be for the male and for the female. So if it's for the male, you would cut the sperm ducts so that the sperm is unable to leave the body. Whereas for females, you would cut the oviducts so that the egg is unable to go and be fertilized by the sperm. Okay, so based off of the table, we can see that sterilization is a lot more effective than the male condom. So we can say sterilization is more effective than the male condom. It is also a permanent procedure. A permanent procedure. However, sterilization, because it's a surgical procedure, is invasive and could lead to complications. Okay, now with the male condom, they're very good at protecting against sexually transmitted diseases. So the male, sorry, I said that twice. Male is effective against sexually transmitted diseases. Okay, question 5 done. We'll continue on with question 6 in the next video.